Good morning, everyone. I would like to welcome all of the resource persons, faculty members, and participants in our 2 days national webinar on significance of paleo environment and its impact in present scenario, organized by Department of Anthropology, Department of Botany, in association with IQAC of Shampoo Siddheshwari Mahavidyalaya. Today, we are going to discuss about paleo environment and its significance as well. Paleo environment is a umbrella term which primarily deals with several disciplines like paleontology, paleobotany, paleoanthropology, paleozoology, paleoecology, and so on. Geological timescale portraying every minute details of paleo environment with numerous eras. Among them, Cenozoic era is most important to understand the story of paleo environment. Cenozoic era is divided into tertiary and quaternary periods. Paleo environment, generally denoted as a climatic fluctuations of Pleistocene epoch, which is inseparable part of quaternary periods. In this time, a drastic change could be observed by means of great ice age and fluviation. Several key elements have been collected to analyze the paleo environment throughout the globe. Sometimes these elements are rocks, pollens, seeds, shells, and huge amount of plant and animal fossils. In a nutshell, this is a study of past environment and interaction between organism and environment. We can boldly say that there is still witnessing the evolution in flora and fauna. Now, without lingering my speech, I would like to start our session. I want to express my heartfelt gratitude to Dr. S.K. Bose, our principal sir, for his endless support and involvement. I would like to request you, sir, to deliver your welcome note in this special occasion. Screen is yours, sir.
Good morning, everybody. First of all, I welcome all the participants in the 2 days national level webinar on significance of paleo environment and its impact in present scenario, being organized jointly by the Department of Anthropology and Botany in association with Internal Quality Assurance Cell of our college. I would like to convey thanks to the honorable speakers of this webinar, Dr. Abhishek Bhomik, Independent Researcher, Anthropology, Dr. Ruby Ghosh, Scientist D, Birbal Sani, Institute of Paleosciences, Lucknow, Dr. Sudha Gupta, Assistant Professor, Department of Botany, University of Kolani, and Dr. Devashish Kumar Mondol, Assistant Professor, Department of Anthropology, University of Calcutta, who will deliver their valuable lectures in this webinar. I would also like to convey thanks to the coordinator and members of the Internal Quality Assurance Cell, along with the teachers of the Department of Anthropology and Botany of our college, who deserve it for arranging such a webinar in this time of pandemic. I think the participants will be benefited from this webinar. I expect a grand success of this webinar. Thank you all once again.
Thank you, sir. I am honored to organize this webinar in association with Internal Quality Assurance Cell of Shampoo Siddheshwari Mahavidyalaya. Moreover, I would like to thank Professor Rajashri Mukherjee, the coordinator of IQAC, and the entire IQAC members. I would like to request IQAC to deliver a speech. Screen is yours, sir.
Thank you. Thank you, Professor Rajashri Mukherjee, for giving me this platform to represent IQAC of Shampoo Siddheshwari Mahavidyalaya. First of all, I would like to convey my sincere thanks to the principal of my college, Dr. S.K. Bose, whose continuous support and encouragement made this program fruitful. My extreme gratitude also for Mr. Kalipodo Mondal, president of my college, and the GB member, Mr. Nudebasi Jana, who are the mentors of this graceful webinar. I would like to thank to the entire team of Botany and Anthropology Department and the convener Dr. Satrupa De of Botany Department and Ms. Soumita Chatterjee of Anthropology Department, who are the real architect of this program. I would also convey my appreciation to all the speakers whose deep discussion on paleo environment will help the student as well as our teachers like, and last but not least, the coordinator of ECAC who gave us moral support, and also I am thankful to the technical supporters of this webinar. Thank you.
Thank you again. Now, without wasting a minute, I would like to request Ms. Shati Sheikh from Department of Botany to introduce our first speaker. Over to you, Shati.
Thank you. Dr. Abhishek Bhowmik has obtained PhD degree from University of Calcutta in 2019. He worked as a researcher in Department of Anthropology, University of Calcutta as Junior and Senior Research Fellow of University Grants Commission. He obtained the MSc in Anthropology, specialization in Archaeological Anthropology from West Bengal State University and secured the first rank. He has a teaching experience of more than 4 years of postgraduate and graduate courses as guest faculty. He has published 3 research papers, and 2 papers are accepted for publication in national and international journals. He has presented more than 15 articles in various international and national seminars. Apart from his ethnographic work on Pauri Bhuiya of Keonjhar, Odisha, he assisted many prehistoric field works for different universities and colleges in Eastern India during his PhD work. Now I would like to welcome Dr. Abhishek Bhowmik to deliver his lecture on Introduction to Environmental Archaeology. Thank you.
Good morning, everyone. Thanks for this introduction, beautiful introduction. Before I start my deliberation, I want to convey my thanks to the principal sir, members of IQAC, the Department of Anthropology and Botany of Shyampur Siddheshwari Mahavidyalaya for giving me this opportunity to present my thoughts in the 2-day national webinar on significance of paleo environment and its impact in the present scenario. I am also thankful to Soumita Chatterjee, the faculty member of Department of Anthropology, for nominating me as the resource person for this webinar. Thank you all. Now I will start my deliberation in the introduction to the environment archaeology. Let's begin with the lecture. So, in the scenario of paleo environment, we are going to understand what is the environment. Environment is the surroundings of an organism, including all the physical, chemical, and biological factors with which it interacts with respect to time and space. So let's understand what is the physical, chemical, and biological factors. Environment consists of physical factors can be of atmosphere, hydrosphere, lithosphere. In the chemical situation, we can understand that it is acidic, neutral, basic. In this one, the biological one can be understood with the producers, which is generally considered as the plants, the consumers, which is generally considered as animals, and the decomposers is composed of the biology. And the culture is the social organization, economic organization, political organism of the organism in the environment scenario is considered as a cultured environment. we can understand the right hand side of this one. I have put forward the biotic factors and the abatic factors. It's very uncommon that all those elements which are having does not have life are called abiotic factors, and which have life are called biotic factors of the whole environment. Let's come into the fact, what is paleoenvironment? The study of environment and its components with respect to time and space in the geological past or in the past. So what are the factors? We can say that again the whole things, when it is composed of the past, it's considered as the paleo environment. Now understood what is about the environmental archaeology. Now considering this paleo environment in the scenario, we can understand that what environmental archaeology is the branch of archaeology which specifically studies the different aspects of past environment to reconstruct the same for human. Environmental archaeology generally can be considered as the study of paleo environments as human habitats. According to the Husserl, we can understand that the core of environmental archaeology is only the human ecology, or can be considered as human mission. Environmental archaeologists employ techniques and concepts developed in the disciplines of anthropology, ecology, zoology, botany, geology, oceanography, climatology, pedology, and many more. So it is about the interdisciplinary studies which help to convey about our studies in the environmental archaeology. Let's understand the aspects of the environmental archaeology. So to make it easy to understand, I have divided this whole lecture into 5 major sections. The first one is the climate and its reconstruction. The second one is the geomorphological aspects and its reconstruction. We can say this one: sediments and soils, vegetation and the fauna. Let's understood one by one in this scenario. So climate and its reconstruction, what is it? Climate, according to the lab, climate is the sum total of weather experienced at a place in the course of years and over the years. So this is actually continuing to the various and longer duration of time. When the weather is considered constant, it is called the climate. Let's understand the spheres of this climate. So considering the environment and the climate, we can consider that 5 spheres of climate are very essential to sustaining the life, as well as to understand this environmental archaeology. The first one is the atmosphere, the dynamic life-sustaining layer of gases and suspended particulate matter that surrounds the planet and distributes the radiant energy of the sun. The Earth's weather is mainly determined by the turbulence within the lower 10 kilometers. That is generally we are considering here in the environmental. This is the troposphere, which is more relevant to the human beings as well as the other organisms. And another one is the geosphere, the solid and the viscous mineral matters of the planet, especially that manifested near the surface as rock and sediment. Hydrosphere, the water on the planet in a liquid form, whether salt or fresh, on with the intersides of the geosphere. This is more important. Whatever is in the geosphere, the water hydrosphere is within that. Now the cryosphere, it is very important to understand the frozen water in the form of ice or snow, usually within a few feet of the surface of the rocks, sediments and water. Is all considered in the cryosphere scenario. And the biosphere, the living organisms of the planet occurring on and in the rocks, sediments, water, ice and lower few hundred meters of the air is called the biosphere. These are the 5 spheres of the planet which we have to understand to make clear the environmental archaeology. Now understand what is the climate of the present scenario. Because until we understand the present scenario, we cannot reconstruct the past. Here, I'm not dealing with all these types of climates and all this. What I want to make here clear is the zones. That the hottest in the equatorial zone, we can say that the hottest region is the equatorial, and throughout the towards the pole, it getting lower and lower temperature to become the sub-zero temperature. It is common in the Arctic Ocean, high ice caps, Greenland, and Antarctica. Now, why I have chosen this? Because it is the Pleistocene environment is considering as the epoch of the human. To understand this epoch, we will have to understood these types of sedimentology and every aspect in the form of this climatic region. So, to understand this one, climatic condition of Pleistocene, because we are considering this as a remarkable epoch in the geological time period of the Earth, marked by the appearance of man and great climatic events. That's why we are considering here on the Pleistocene epoch, because it is generally considered as to be human. The most widely referred glacial and interglacial cycle is that recorded in the Alps mountain. These sequences in alpine region have been identified by two geologists, Peng and Bruckner, in 1909 on the basis of their studies of ancient sedimentary and other deposits called moraines made by the glaciers during the sliding movement. We are going to understand the moraines in a very detailed manner in the next session where sediments are there. So you can, in the right hand side, there is a GIF photo is there where you can understand that it is continuously, the zone has been declining, and throughout the years, it is from the 21,000 years to the 1,000, it is showing there that how the means the ice capping is reducing day by day. And when it's in 21,000 years before, what was the scenario, and now what is the scenario? Thus, the continuously in the Pleistocene era, there is a continuously growth in the scene of the alpine regions, and these one as the growth of the whole ice capping in the planet. These are, and there is a continuously retreat also. With the help of these things, we can understand that there are four types of glacial age: Gunj, Mendel, Riss, and Fold, which are intervened by the three interglacial periods, namely Gunj Mendel, Mendel Riss, and finally the Riss Fold. Now, if this is in the alpine region, what is in the equatorial region? In the climatic condition of Pleistocene, we can understand in the equatorial region, we can there is a tropical region of the Earth experience high precipitation and high humidity in some regions of the tropics due to high precipitation rivers and streams were saturated with water, causing intense floods and sedimentation. These sediments, deposits in the form of silt, sand, and gravel, were formed extensively in the floodplains of the river in the tropics. These period of flood were termed as pluvial, and the period between the two pluvials were named as interpluvials. With the understanding of many of the researchers and articles, the four pluvial periods were traced on the basis of geological evidences in the East and South Africa, and these are namely considered as Kageran, Kamashian, Kanjiran, and Gambian. Let's have a look on the geomorphology. This is all about the climatic condition. Let's understand the geomorphology. Section 2. The geomorphology can be easily considered as the landforms and its studies. Reconstruction of past landforms and surfaces is an essential aspect of environmental archaeology because the spatial context of a site is crucial to its interpretation and to understanding its relationship to other sites. Space and landscapes define the resources available to any human group, and the landform changes through time are related variously to changes in the other elements of the environment: climate, hydrography, flora, and fauna. Examples. For examples, the alien landforms. How it is considered, means it's been created, is the help of the air. Some kinds of landforms have been created. For an example, sand sheets, dune fields, loess, deposits may dominate the landscape, blanketing earlier landforms. Severe landforms, floodplains and terraces, river channels, and channels forms are the important elements of archaeological landscapes. Even the glacial and glacial fluvial landforms are also very important in understanding the environmental archaeology. Landforms created by glacial erosions and deposition. For just an example, I am going to give a detail of glacial landforms. Here in this picture, you can easily understood that there is a V-shaped valley. It is considered a pre-glacial topography. After the glacier is being there, then this V shape has been changed to the U-shaped valley. It is very common, and why it is important to understand is that apart from this landform changes, every aspect related to these, the shoreline and everything has also been continuously changing. So it is very important to understand the landforms and geomorphology of an area and a site to make it understandable for the archaeological perspective. Let's understand the sediments and soil. So, in understanding the sediments and soil, what are sediments and how it is used? Sediments are composed variously of particles, disaggregate rock, dust from whatever source, bits of dead animals and plants, and chemical precipitates. So everything constituted as a sediment. Sediments form when particles in transit are deposited. As transport medium lose energy, the conditions of transport and deposition can often be inferred from the attributes of sediments, which are important source of proxy data on former environment. We are actually reconstructing the environment. So these sediments are very useful in reconstructing the environment. Sediments constitute the context of archaeological remains and sites, locking them into the stratigraphic and locational relationships with other classes of data. And sites in the primary context may be buried by the aeolian, alluvial, colluvial, volcanic, marine, or lacustrine sediments. So, how it is useful actually when we find some of the artifacts or some of the useful informations buried under the earth, if these are there, then we can easily understood that which article or which artifact is present in the which sediment and how it is being transformed or it maybe has transferred to from the other places. This can also be understood from this type of studies of the sediments. So the deposition of the glaciers is very essential to understand all of the environmental archaeology, and it's very well known that it has the most founding effect in the human history. So what types of depositions are done by the glaciers? These are drift. Is any material deposited by glaciers or other meltwater? Till is that unsorted material that is deposited directly by the ice. Moraines are the linear feature deposited at the bottom or along the sides of the glacier. And glacier erratics are enormous boulders transported and deposited by the glaciers, often far from their source of origin. I cannot have this all of the data to be created by me, and you find the some of the photographs, but I have two photographs where we can understood about the tilt plane, terminal moraine, and outwash. So how it is being prepared? You can see in the left hand side of the figure, the glaciers are coming and then the glaciers are retreated. Now what we can have is the tilt plane there, the sediments which are tilt, the sediments which are terminal called as terminal moraines, and the outwash plane where there is no evidence of the means actually that becomes the water and it outwashes the all of the sediments. So, and in the right hand side we can understand about the lateral moraines. Those moraines which are present on the both sides of the banks of the glaciers are called lateral moraines. Recession moraines are those when the glaciers retreat, it leaves us the means block of these moraines. These are called recession moraines and the terminal moraine up to which the glaciers has extended. So we have understood from the previous lecture that previous slides that what is there, that it is retreat and this the Grunge in the Grunge scenario, the ice sheets are extended to a certain landmarks and then it retreated. So these types of evidences from the depositions of the glaciers we can understand these things. Okay, so let's understand with the soil. Soil, the science of soil is considered as a pedology, is the chemically and mechanically altered terrestrial sediments. When it is chemically and mechanically altered terrestrial sediments, it is called the soil. In archaeology, the soil, its layers, are the fundamental source of information, which is generally known as stratigraphy. We generally have this soils, if considered as a topmost soil, then it not only the topmost soil, but the layers is considered as the archaeological source, and different types of soils from the different strata, and as the soil is indispensable part of the environmental archaeology. In the stratigraphic section, interruption in texture, structure, or mineralogy of the sediment columns are the criteria for defining sedimentary bedding units. Comparison with the sedimentary sequences beyond the immediate locality are aided by the reference to the formally defined hierarchy of lithographic units. Lithographic units are defined and described at type localities that are standards of comparison and formal naming. So strata, how to name these stratas, it is been discussed here. Because of the naming and making it a different shape, we can understand the layers in a sequence manner. Let's come into the vegetation. So what is included in the vegetation? The organisms most central to environmental archaeology are eukaryotic. Means we are dealing with the eukaryotic, those having multicellulars. So plants, some protists, occasionally, and some fungi are the parts of the vegetative fauna which is being used by the environmental archaeologists. And what types of data can be considered? What is the source of the data? So we can understand this one as a three classes. The macrobotanical remains, that is visible and recognizable trees on parts of plant. Macrobotanical remains are pieces of wood, seeds and fruits, stems and roots, leaves, buds, cuticles and so on that are preserved under diverse special conditions. Microbotanical remains can be considered as plant parts and products requiring magnification for the study. These are actually called microbotanical parts. Mostly microbotanical remains include principally pollen and spores. Microorganisms such as algae, diatoms, inorganic intracellular deposits such as open phytoliths and calcitic crystals are extremely important in not only understanding the environmental archaeology but also the paleobotany, where we can understand from various sources that how the environment is there and which type of vegetations are present in the concerned environment. Molecular and chemical residues and traces taken out with the sediments, charred crusts, or animal tissues in direct evidence for past plant association. Habitat groups. The species distribution comes from such proxy sources as sediments and soils, animal remains, and for recent studies, artifacts such as graphic representation and historical text. This is actually the animal remains and the recent studies of artifacts such as graphic representation and historical text is that when we find some of the graphical representations in the artifacts such as pottery and something which is relevant to the study of archaeology, we can understand that it is the way we can put forward our thoughts, and in considering these thoughts we can put forward that how we can make it out that which type of climate, which type of vegetations are there at the time of which manifest in artifacts. Some of the consider if we consider about some of the means, cave paintings and there if we find these kinds of remains we can understand that what types of plants are remaining in sources. So this is also very important source of understanding of the vegetation. Let's come to the fauna. Why do we study the fauna? Why we study actually fauna? Humans and other animals evolved together for over 3 million years in Africa, in Northern. Faunal remains in archaeological context support inferences about the environment of the animal themselves, climate and habitat, as well as aspects of human environment that animals affect or reflect. Interpretation of animal locations and season of availability are important aspects of paleoenvironmental research that are widely honored in the study of archaeological animal remains. They directly implicate human behaviors in animal exploitation, particularly hunting and pastoralism. So that's why not only understanding the paleo environment, but how they are associated with the humans is also can be understood by understanding the fauna and their remains. For paleo-environmental research, interest in faunal remains cannot be restricted to only the large vertebrates or only bones. The full range of animal remains in the archaeological context can be informative, particularly about the ecology, preservation conditions will control the availability of animal tissues in any form. Whether archaeologists encountered bones, antlers, horn, chitin, hair skins, and other tissues, feathers, birds, eggs, or mollusks, shells, parasite crates, or chemical residues. So, different types of faunal remains can be attributed to different types of behavior, understanding of the behavior of the human beings in the past. The full range should be recognized, sampled, and treated approximately to establish conditions for successful analysis. So, before what understand is that if we consider the whole of the Pleistocene epoch is identified by the occurrence of Villafranchian fossils, namely Bos, Equus, and Ficus. It is actually what we understand is that Villafranchian fossils are very essential to understand the Pleistocene and Pliocene boundary, where there is boundary considered from where we can understood that what type of climates and other things are there and what is primitive. So these are the faunal remains which can decipher much more information rather than only the environment but also how human is going to adapt this situation in his past behaviors. I think this is my all what I want to say in this whole of the lecture, and I just want to say thank you for patient listening. That's all I have to say. Thank you.
Thank you. Thank you, Dr. Bhomik. Actually, there is no such a question. And thank you, Dr. Bhomik, for such a nice presentation. A single question has been derived, that is, what is lacustrine sediment, from our participants.
Okay, thank you. So, what is lacustrine deposit? Well, the deposits are present in the lake and means lakes. If we study the lakes in the paleo environmental situation, then there are some of the deposits that are produced by the lakes. These are actually considered the lacustrine lakes. That's all I have to say.
Thank
I
you,
think
sir.
I can make it good. Okay.
Thank you. Now I would like to request Ms. Rituparna Raichaudhuri to introduce our next speaker. So over to you, ma'am.
Shomita, I think there is some problem in Rituparna's end due to lack of network. So can you please introduce the next speaker? Shomita is not there in the stream right now. Sorry, Rituparna is not there in the stream right now. So can you please do that?
I need a help from Shatarupa De, our next convener.
Thank you, Shomita. Now I would like to introduce our next speaker, Dr. Ruby Ghosh. Dr. Ghosh is currently Scientist D in Birbal Sahni Institute of Paleosciences, Lucknow. She did her PhD in 2008 from University of Calcutta on archaeobotany. Her research interest includes vegetation response to past climate change and understanding the drivers behind the change in the ecosystem. She is the recipient of several awards and honors, such as Birbal Sahni Research Scholar Award in 2002, Pratul Chandra Bhandari Medal 2006 from Birbal Sahni Institute of Paleosciences, Lucknow. She was awarded INSA Visiting Fellowship for the year 2012-2013 and selected for INSA DFG Bilateral Exchange Program in 2016 to work at renowned Senckenberg Research Institute, Frankfurt, Germany. She was also awarded Excellent Scholar Award by International Botanical Congress held at Shenzhen, China in 2017. She has published several papers in journals of international and national repute. It is my pleasure to welcome her in this webinar, and I would request her to start her talk. Over to you, Rubidi.
Thank you, Shatarupa, for this nice introduction. I don't know whether I deserve this or not. Well, thank you. And I want to thank all the team members and the principal of your college, that is Siddheshwari, Shampoo Siddheshwari College, and for giving me this privilege to talk over here. And I also want to welcome you all to the world of phytoliths, and I just want to begin my lecture. Okay, today I am going to talk about a tiny bioclimatic proxies and its implication in paleo-environmental researches. And before going into details of this, I want to let you know why our past is so important to us, because past gives us lesson to cope up with the future and to for betterment our present situation, such as you all know that nowadays we are all threatened and by a pandemic, COVID pandemic, but it is not new to the mankind, because 1918 was also people of 1918 are also seen this kind of pandemic when above 500 million people were affected all over the globe. And if we should have taken the lesson from this past, so we could have minimized the casualties right now which are facing nowadays. So past is, in a brief word, past is the lesson for us, so which can better our future. So past climate is very important to us, because if we know the trend, how the climate is changing over the days or over the years, we can, if we know the change better, so we can predict the future climate trends, and we can mitigate the effect of the climate change impact on the mankind as well as on the ecosystem. So paleoclimate is so important to us. And now the paleoclimate proxies, I will just give you a glimpse about the conventional paleoclimatic proxies like pollen and spores, which are being long used as conventional paleoclimatic proxies, wood ring studies, stable isotopes, paleothems, all are these very important paleoclimate proxies which are being used all over the globe, which are being used by the researchers of all over the globe. And pollen and spores are, help because these tiny microgametophyte and asexual reproductive structures of the planorgams and cryptogams have very unique structure. Their shape, size, ornamentation pattern, and aperture provide them uniqueness. And with this uniqueness, they help us to identify up to genus, species, or level of the plant, so we can reconstruct the vegetation from this tiny bioclimatic proxies. Since long we are using this, they are very efficient, and this porous pollenin in the wall of this pollen grains or spore give them resistance against physical destruction during their preservation. Okay, now the wood ring are also very important bioclimatic proxies. We know that this plants produce these rings in response to climate change, and as a response of climate change they produce wood rings. And in a year they produce two kind of rings, like rings which are known as early wood, produced in the spring season or early summer season, or summer season wood, they are known as late wood. Okay, if we and these are very important, not for not for understanding the chronology or growing span of this plant, but for the paleoclimatic perspective also, because they also retain the climate change impact within themselves. So they are very useful and high resolution proxies, I must say. And another important proxies are stable isotopes. Stable isotopes are very useful for reconstructing vegetation as well as climatic condition over past. And speleothems are also important proxies. These are found in cave deposits. Probably mostly they are made up of dolomite or calcium carbonate deposits within caves, and they are also high resolution climate proxies if we perform oxygen isotopic studies or carbon isotopic studies with them. And so these are very important paleoclimatic proxies. So but all these proxies have some demerits, like pollen and spores. We know they are very reliable proxies, but still in the fluvial deposits or in the oxidized condition, they are rare. They are of rare occurrence. They are of rare occurrence because this sporopollenin gets destructed in the oxidized condition. And wood ring studies, although it is very high resolution, but it doesn't give us long-term result, because the longest tree ring result is about 7600 years ago report. And the stable isotopes can identify the source only, but it cannot tell whether these source are from terrestrial plant or if it is it can identify the terrestrial plant, then it cannot discriminate about whether it is a herb or shrub or tree, etc. And speleothems are also very important, but still we have to find them in caves, and you won't find caves everywhere. So we need another proxy. We need some other proxy where which can overcome these demerits. Let's see what phytoliths can offer. So why we should study phytoliths? Because phytoliths have advantage over pollen grains, because they are siliceous in nature, and they are very resistant to degradation in oxidative condition or natural stress or any taxonomic bias. They are resistant to taxonomic degradation. And also they have advantage over pollen grains because in the pollen with the pollen grains we can identify whether it was a forested environment or it was dominated by grass, whether grasslands were there or not. But pollen grains cannot identify if the grasslands were dominated by C3 or C4 grasses, because grass pollens are very similar in appearance, and but phytoliths have advantage in this, and they can identify whether grasslands were dominated by C3 and C4 grasses, because we know this photosynthetic pathways are also responsible for their ecological differences. So they can offer, and among the C4 grasses also they can discriminate the warm humid loving grasses and warm dry loving grasses. So these are the advantage of this phytolith study. Now, just a brief idea about what phytoliths exactly are, and where to find them. Phytoliths are made up of silica, as I said earlier, and these are practically plant opals, amorphous silica with H2 in them. And how they are formed within plants, because this silica is found dissolved in groundwater as monosilicic acid, and through root hairs plants intake silica within them, and through transpiration pool these phytoliths not phytoliths at that time, they monosilicic acid travel upwards in the plants, and get deposited within intercellular spaces or even within the cell sap also. And once it is once it has penetrated the cell sap, it started form dimers and oligomers, and after that it polymerization is there, and with due course they form crystal, and these are very hard crystals. And after the death and decay just have a look, this is a schematic representation of the grass epidermis, where long cells are there as well as the cork cells are there. If this silica are precipitated within the elongated cell, and then after the death of and decay of this plant tissue, they will retain the exact shape of this cell. And if this is penetrated or deposited within these cork cells, they will produce cells like smaller size cells, similar in shape and appearance to the cork cells. After the death and decay of the plants, this silica are exposed in the and dispersed in the soil, and after some time on they started settling down, and with due time they will get fossilized. So after fossilization, when we recover them from any fossil sediments, they give us clue about the plant where they were originated, where sometimes they can identify family, sometimes they genus, and sometimes even species also. Mostly we still focus on the assemblage analysis, not individual silica morphotype we care more about. Now, uh, apart from this paleoclimatic implication, these silica have some functions within the vascular plants. And what are they perform? Or what they perform actually? These silica, as you know, these are silica are very hard, so they provide strength, mechanical strength, and they also lessen the effect of the radiation damage, and provide resistance against the pathogenic attack to plants, and as well as herbivory. They also lessen the herbivory, because silica are coarse in nature, so herbivores plants rich in silica, herbivores don't like. So these are a few functions of phytoliths in vascular plants. There are many more, so I am not going into detail. Now, where to find these phytoliths? Actually, phytoliths are found in also bryophytes, also in algae. That means cryptogams are also having phytoliths, but we'll more concentrate on the seed plants or vascular plants like pteridophytes and gymnosperms. Being a cryptogram, pteridophytes, 11 families of pteridophytes are known to produce phytoliths, and in gymnosperms also 11 families are known. Monocots are diverse producers of phytoliths, 28 families known, and of this, Poaceae, Cyperaceae are the dominant producers of phytoliths. Dicots, among the dicots, 103 families are noted till now, and this still increasing every year. This counts are still increasing. Okay. Now, collection of sample, depending on the objective of your study, you have to collect your sample like from plant living plant, surface soil, subsurface sedimentary profiles, and there are different methods for extracting these biogenic silica from these plants and sediments. And after getting this, we have to study them rigorously, and after rigorous study and statistical analysis, we can interpret this data in climatic terms, and then I will give you a glimpse on how this can be done. Now, these are the lakes, peat deposit, ditches, soils, ice or glacial deposits, cave deposits, archaeological deposits are best place to find phytoliths, and they can be used in solving the query related vegetation, climate, etc. of the past. Now, as I show, as I showed you earlier, that phytoliths, on depending on the plant tissue where they are precipitated, they retain this particular shape. And phytoliths are can be broadly classified into two categories: grasses and non-grasses. And grass phytoliths are of two types: short cell grass category phytoliths and elongated morphotypes are there. And in both the cases, for grasses and non-grasses, they may be dispersed or articulated phytoliths. And I just want to mention here, these short cell morphotypes are more significant for us because they have ecologic significance, ecoclimatic significance. I will let you know how they give us clue about this ecoclimatic condition of past. And elongated morphotypes are not taxonomically or ecologically most important things, but only the bulliform cells, they give us some result. Okay. Now, these are the light microscopic photographs of some phytoliths. These are mostly grass phytoliths, and I will show you how these are important. Just look at these dumbbell shaped phytoliths. These are all bilobeds. These are known as bilobeds. These are trilobids, these are quadrilobids or crosses, these are polylobeds. This type of lobate morphotypes are more abundant in the members of Panicoideae subfamily of Poaceae. Okay, first I should mention here that there are 12 subfamilies of Poaceae, and among which Panicoideae is the warm and moist loving grasses. Members of Panicoideae are warm and moist loving grasses. They are mostly C4, but few C3 members are there. So if we retrieve this lobate types more in the sediment, we can say that okay, these Panicoideae grasses were dominating during the time of deposition, and corresponding climate was warm and humid. And these are called shaddle. These axe-shaped morphotypes are called shaddle. These short saddles are characteristically produced by the members of Chloridoideae. They are also C4 grasses, short grasses. They are C4, but they are adapted to warm and dry climatic conditions. So if we recover shaddles in abundance, so we can say that okay, the climate was warm and dry, and Chloridoideae members were dominating the assemblage. And this type of long shaddle and collapse shaddle types, these long shaddle and collapse shaddle types are chiefly produced by the bambusoideae members, the bamboos. Generally, they are C3 and they love warm and humidity, you know. And as I said earlier, the elongated morphotypes are not much helpful for taxonomic or ecologic purpose, except the bulliform cell. This fan-shaped bulliform cells, these are two also bulliform cells. The most important thing is this fan-shaped bulliform cells. We know that nowadays it is known as bulliform flageolet. When this bulliform are produced, bulliform cells are produced in higher number when plants are water stressed. In response to water stress, grasses started rolling their leaves, and to cope up with the situation, they roll their leaves and produce more bulliform cells. So, if we get a higher number of bulliform cells in a sediment, so we can say that there was water stress in that condition. Okay. Now these are called rondelles. These are called rondelle morphotypes, and these are chiefly produced in the members rondelles and tapidiforms. These are mainly produced in the members of Puoidae. Puoidae are C3 grasses. They are full loving grasses, and also they also love high or available soil moisture. So they are adapted to the high elevational zones like mountainous, higher mountain. Okay, and these hat shaped structures are produced chiefly in the sages, and these are pardon me, these are not my picture. I have taken this from some literature. These are globular granulate morphotypes chiefly produced in the woody dicot, that means forest indicators. And this multifaceted type is chiefly found in the cucurbitaceae, and this globular morphotype is spinule are noted in the palmy family or palms, and this blunt hat is found in the hibiscus. And these are all the globular members are recovered in some plants which are adapted to the high salinity zones, that is mangrove plants. And this was a publication by our group in 2013. Now, before giving you an idea about how this phytoliths are helpful in reconstructing the past environmental condition, I should just focus on briefly about another function of the phytolith research. Another implication of the phytolith research, it is more important because understanding of the plant domestication, it is related to the human society. So I thought that I should give you an idea about this. And I have just selected 3 cereal crops here, and how phytolith can focus on the plant domestication. Look at this. These structures, these horn-shaped structures are called uh, double peak bloom cells. These are actually recovered from the rice bloom, both in domesticated as well as in wild rice varieties. So this group of scientists, Zhao et al., they have studied a large number of Chinese varieties of domesticated and wild rice, and as well as other Asian rice varieties. After studying these large varieties, they took these measurements like the central depth of this, height of this tower height, middle height. These measurements they have taken for all these varieties they have considered, and um, they propose two discriminant function, one for domesticated rice and one for wild rice. If you put this measurements into these functions, you will have an answer. If this prediction of this the function for domesticated rice gives the number, if it is higher than the wild rice, then you can be assured that the rice you are getting, rice bloom cells you are getting, is pointing towards the domesticated rice. So in this way, you can say when exactly the plant domestication has started in the past. And similarly, this bulliform flagellator also very important, because wild rice and cultivated rice, both of these varieties produce this bulliform cells. But in case of wild rice, these are smaller in size and with lesser number of ornamentation. And in case of cultivated rice, this is larger, quite bigger in size, and with a large number of ornamentation, generally more than 14 in case of cultivated rice, and in case of wild rice it is always between 9 to 14. So this way also, if you don't get this bloom cell, then also this bulliform cells can help you in identifying the rice domestication phase. Now another important crop, the maize. It's very important for neotropical countries, and it also originated in neotropics. And this group of scientists just studied them, and the wild ancestors of maize also they studied, and they also studied the domesticated varieties of maize, and the some wild grasses which resembles maize. And after studying, they get this type of phytoliths which may be used in distinguishing these different varieties of maize. If we find this ruffle top rondel, these are called ruffle top rondels. The rondels will frill in the top. If we get a ruffle top rondel in abundance, we can say that the genus Gia may be present at that time. But in addition to this, if we get also this rondel with wavy top, this wavy top rondel is the indicator of Gia maize. That means the cultivated maize. So if we get this, we can say that confirmedly, with these two morpho, we can say confirmedly Gia maize was cultivated at the time of deposition. And this type of half decorated rondels are the dominant character of the ancestors of the domesticated maize. So like this, we can identify also the maize cultivation when started in the neotropics also. And another important crop which is growing, the Setaria italica or foxtail millet, which is found in Eurasia, and it has originated in Eurasia. There is a long-standing debate. Okay, now how to differentiate between the foxtail millet and green foxtail when you get only the churd remains? It won't help you. It is morphologically very similar. So in that case, you have to take out if you get the carbonized remains, not churd, you have to take out this lemnar pelia bloom cells or the Haas type. And in case of this, you will find that the Setaria varieties or green foxtails are producing smaller crosses in compared to the Setaria italica, the foxtail millet one. And similarly, these omega shaped ornamentations are there on their husk, and for this omega 3 type ornamentation it is a discriminating factor for foxtail and green foxtail millet and green foxtail, and because these are larger in size and number for case of the foxtail millet. So this can be helpful for this also. So this was a glimpse and how phytoliths could also be used in identifying the plant domestication practice in the past. Now come to our point. That is my focus is the reconstruction of paleovegetation and paleoclimate using phytoliths. So this was a very young. When I was young, it was done in that time with my group, and actually these are the coprolites. These coprolites were collected by Professor Shubir Beda, who is my mentor. He collected this from Yanghai Tob of Xinjiang province of China. These coprolites were dated back to 2,500 years BP. So they captured the climatic phase of particular that time, and we recovered a diverse phytoliths dispersed as well as articulated the sea, and we found that it is we recovered short saddle in abundance and as well as these rondel types in abundance. So if we recover these rondel types, rondels are indicators of poetic grasses. That means a cool climatic condition was there at the time of deposition, and and we also got the rondel morphotypes, short saddle morphotypes in abundance. So it indicates that during the time of deposition it was drier. So a cool, dry climatic condition was inferred from this phytolith study during the time of deposition in Xinjiang province. Now, how phytoliths also can help us to identify the different deltaic sub-environments. We know there in deltaic mud flats, different sub-environments exist with the changing salinity, and mud flats near to the coast are high with high salinity. This zone is high salinity zone, and if we go landward, salinity will decline gradually. Okay. Now we have collected 50 grasses covering all these 5 subzones of the deltaic environment, and we noticed that on the basis of phytolith spectra of these 50 grasses, we noticed that swampy mangrove, the true mangrove zone, and landward zone were I mean to say the zone which is high in salinity and with low salinity zones can be discriminated on the basis of phytoliths. As you see here, this type of tower shaped morphotypes are produced in high percentages in the grasses which are collected from this true mangrove environment. But the grasses which are collected from the landward direction, that means low salinity or freshwater condition, uh, they produce high percentages of bilobates and process. So on the basis of this, we can discriminate the different sub environments of the mangroves also. And for this study, we had considered our Sundarban mangrove chunk, Indian part of the Sundarban, and later on we thought that if grasses can work, why not non-grass? So we started investigating the non-grass, and we collected about 33 non-grass covering the both mangrove plants and mangrove associates, and covering these 5 subzones, and we found that globular morphotypes are dominantly produced. These globular morphotypes are dominantly produced in the mangroves, whereas the mangrove associates or plants collected from the mesophytic zone, they produce the hair cell base or anticlinal plates or other morphotypes which are not evident in the mangrove plants. So on the basis of this investigation, we say that we can discriminate if we found in a fossilized sediment from this Sundarban delta, we can discriminate the environment of deposition of that stratum. Now come to till now I was talking about the phytolith assemblages, how these phytolith assemblages can help us to reconstruct vegetation as well as climatic condition. But in that case, we have involved both long cells and short cells morphotypes. But here, for environment analysis, we will focus only on the short cell phytolith morphotypes, generally produced by grasses, and some non-grass also will be there. For example, it is D by P degree. This is the D by P or tree cover density index, originally proposed by Alexandre et al. And Bremond et al. Just revised this later on. This is the proportion of the globular granulate and other short cell morphotypes produced by the grasses. If it is greater, if this index shows that values are greater than 1, that means during the time of deposition there was a forest nearby. And but if it is lower than the values are lower than 1, it suggests a grassland condition and dominance of grasses. But it cannot portray whether the grasses are C3 or C4. That can be done by this IC or climatic index. It is the proportion of the poid and chlorioid and other C4 grasses, so that we can have an idea whether during the time of deposition in the past the grassland was dominated by the cool loving or warm loving grasses, and that is the C3 or C4 grasses. Now further, the C4 grasses can be discriminated whether they were warm and humid loving or warm and dry loving grasses on the basis of this IPH, which is the proportion of this chloridoid and panicoid types. So these three indices are very helpful for paleoclimatic research, and being used by many researchers all over the globe while using phytolith indices and assemblage in environmental reconstruction. Another important index is FS. As I said, that this percentage of fan-shaped bulliform cell should increase with the increasing water stress. So if we get higher number of bulliform cells in sediment, so we can say that water stress were there during the time of deposition. So we can likewise we can draw a picture of the past environmental condition. So let's see. So this is a schematic diagram. When IPH increases the xeric condition, it suggests the xeric condition. When it decreases, it suggests the mesic condition. When IC index is high, it shows with the dominance of C3 grass, and when it is low, it shows the dominance of C4 grass. Similarly, as I said, FS index, when it is higher, it suggests the water stress. When it is low, it doesn't suggest any water stress. It suggests that okay, condition was more adjustable for plants. Okay. Now, like other paleoclimatic proxies, I have just told you the strength of phytolith research, but still they have some demerits. Some weaknesses are also there for phytoliths also, and we have to overcome this. To overcome this, we should know the weakness first. So what are these weakness? Weakness are they have some over or under representation issues due to their genetic or edaphic controls on taxonomic bias, and the most important thing is redundancy and multiplicity. I just want to explain what is multiplicity. Multiplicity means production of more number of phytoliths, production of different varieties of phytoliths in a similar in a single taxon. And redundancy means one single morphotype could be present in more than one taxon, but they may be unrelated also. So these are the problems, and we have to minimize this. And I will give some examples from our work. But before going into details of our work, where did we get an idea to proceed further? We should describe that first. In 2008, this paper was published by this Bremond et al. And they studied the two East African mountain ranges and one East African lake, and they covered from tropical to 4,500 meter elevation, and they collected about 25 number of surface soil samples, and they found that and they also had the botanical data in support of their surface soil data, and they found that with the rising elevation, this IC index increases. That means what I said earlier, that if IC increases, it suggests the dominance of C3 grasses, and that C3 grasses are cool loving grasses. So if IC increases with the elevation, then it indicates that cool loving grasses are rising with elevation, and warm loving grasses are restricted to lower elevation zones. And so the IC has a linear relationship with the elevation, and they found the coefficient of determination was quite high, that is about 0.92. So it is quite high. Let's see. Another report came in the 2015, and these authors have collected about 41 samples from the Tibetan Plateau, and this Tibetan Plateau and southern Himalaya, they have collected the samples, and they covered above 5,000 meters elevation, and they collected these 41 samples covering 5 vegetation zones, starting from tropical to alpine zone vegetation zones, and they found that on the basis of the phytolith spectra, these samples can be discriminated, and above 95% of the samples could be discriminated on the basis of phytolith assemblages, and DA also supported their result, and they've also performed some multivariate analysis like RDA, where they have used altitude as the environmental vector, and they found that altitude is the most influential factor for this difference in the phytolith description. And they also assess whether IC index or IPH index, which of these index is working in the Tibetan Plateau. They found that IPH is not the appropriate index for the Tibetan Plateau, which is not working properly. But in case of IC, they also found a very strong linear relationship, and the coefficient of determination was 8.8. So that means 80%. Okay. All these cases from East Africa to Tibetan Plateau, they showed that IC has a linear relationship with the elevation. Okay, it's fine. And very strong is also. Now, this IC, I should mention here, this IC index and IPH index, these indices are being used by different researchers all over the globe, for their phytals research without validating their regional efficiencies. So they have not checked ever whether in their region they are efficient or not. Without checking their efficiency, they started using this IC index. Now, some question are there in our mind, that are they equally efficient in the Himalaya for paleoclimate reconstruction? And which of these indices, there's like which of these IC, IPH or FS, which of these indices is the best for the Himalaya? And now we have to also find out the principal climate driver. All the studies were telling that okay, IC is rising with the Himalaya. It has a linear relationship with the Himalaya, but they don't tell whether which of the climatic driver is influencing this IC. They are not telling this. So we had to find out that. Let's see what our study can portray. Keeping in view all this question, we started our study from two sectors of the Himalaya. We collected about 66 dominant grasses from Darjeeling and Arunachal Pradesh, as well as 147 surface samples were there in our study, and we found that among these three indices, only in our case also, surprisingly, in our case also, surprisingly, IC is working only, and IPH and FS are not working. Okay, let's forget about these two indices. Now concentrate on the IC. But surprisingly, our IC is, although it's linear, showing linear relationship and significant also, but it is not stronger as seen in the southern Himalaya or Tibetan Plateau or in the East Africa. So we started calibrating this index, and we studied all the living grasses of the Himalaya we have collected earlier, and we found that in case of Himalaya, the grass composition is significantly different from that of East Africa and from that of Tibet also. And so we have added some dominant phytolith moths which are produced by the tsetse grasses exclusively in the Himalaya to this, and in this way we have calculated this conventional IC in three different ways, IC, HIC1, HIC2 and HIC3, and we found that HIC2 is the most suitable one for the Eastern Himalaya, but we have to go further. Next, in the last 2 years, we have worked on worked on a large number of samples, and we have included also samples from another Eastern Himalayan sector, that is Sikkim, and also we have included the samples from Ladakh Himalaya, the most rainfall deficient region of the Himalaya. So to check whether rainfall variation have an effect on the IC or not, or how our IC could be improved, so that in the Himalayan region the most accurate result could be found. Okay, this is the temperature and precipitation variability in these four sectors from where we have collected samples. These are our study area. Look, these are the alpine or subalpine zones of the Himalaya, as well as we have temperate and tropical, subtropical zone also. Mm, so this is the schematic representation of the grasses along the Himalayan mountain, rising elevation in the Himalayan mountain, and we found that on the basis of the surface soil sample. In this case, we have included 247 surface soils collected from these four Himalayan sectors, and on the basis of this finding, we noticed that these panicoidy members are panicoidy and chloidoidy. That means the C4 grasses declining upwards. That means with rising elevation they are declining, and in contrast, poidy members are increasing increasing with rising elevation. There are C3 gases. Mistakenly, it was mentioned here as C4. Just it will be C3. And Bambusoideae and Oryzoideae somehow also showed a declining trend, but Orondinoideae and Microideae, they are chiefly C3 grasses, but they are significantly recover from all the elevational zones of the Himalaya. And when we performed the PCA, we found that on the basis of our grass phytolith spectra, this sample could be differentiated as per their zone of collection. And now we have to now the another objective of our study was to find out the principal driver for this grasses, the which influence the phytolith distribution and the C3C4 variation along the elevation zone. So keeping in mind that we included 6 drivers in addition to mean annual temperature and mean annual precipitation, we included mean precipitation of coldest quarter, mean precipitation of warmest quarter, mean temperature of warmest quarter, and mean temperature coldest quarter of the Himalaya in our study. And we performed an redundancy analysis, and ultimately we screened out because on the basis of the background statistics are not going. I don't want to bore you all with this statistical details. So just keep in mind, we found that these principal factors are there which are influencing the IC variation or the gas C4 variation, vital spectra variation. And above of this, the MAC is the most influential one. MAT is found to influence more than mean precipitation of coldest quarter. So further, we took MAT only for our analysis, and now we also had to check how whether why Himalayan ice is showing variations. Why Himalayan ice is showing the variations with the other ices observed in the eastern Himalaya, Eastern Africa or southern Himalaya. We found that in these cases, the published in case of published records from the Eastern Africa or southern Himalaya or Tibet, they have a long variability of the IC range. IC varied from 0 to about 99. Okay, but in case of Himalaya, it is not varying like this. It is quite high. It started about 20 from 25. It varied around 92 or 93, I guess. So the variability is less in case of Himalaya in compared to the southern Himalaya and Tibetan Plateau, southern Himalaya and Eastern Africa. And if we compare the conventional IC calculation on the Himalayan sample with the revised IC, I just want to mention here that we have included 2 more C3 grass indicator morphotypes here after studying 101 grasses from from the Himalayan arc, and we included 2 more C3 grass indicators here, and we found that the revised IC or RIC increases. At least the performance improves. Okay, so we propose to linear model. We built 2 linear models using one conventional IC and the revised IC for the Himalaya, and we found that for the Himalaya, this revised performance of this revised RIC or is better. And we selected the model 2 for Himalaya after considering the validation statistics. It is in background. So now we have to check also the potential of these models for past temperature reconstruction. So we applied these models into the phytolith data of an earlier published record, which was earlier published from the Eastern Himalaya by our group. And not only phytoliths for this profile, this was a sedimentary profile collected from the dried lake bed in the 6 mile of Darjeeling area, in the temperate zone of the Eastern Himalaya. And not only phytolith, this data was complemented with the stable carbon isotope as well as grain size analysis data, and pollen data was also there. On the basis of this, all this data and phytoliths also, we inferred 4 climatic phases for this profile, which covered the last 2,400 years history. And we found that the initial phase was moist, which is which was later on it was a less humid phase, and then comes the strong monsoon, and again monsoon weekend in the towards modern day. Okay, now on this data we have applied these two index, that means the conventional IC and our revised IC, and on utilizing these two models we have reconstructed the paleo temperature variation, just and we compare our data with the other data like the stable isotope data of the profile and non-annual non-arboreal pollen and arboreal pollen data, as well as the carbon dioxide data, PCO2 data of the past for this 2500 years, taken from O'Hare et al. And the map data we have reconstructed using the using Basu et al.'s equation. So we found that in both the cases this MAC reconstruction is following the similar trend, but in case of the conventional IC, all along the MAC is below the present day temperature, so it's not going to work. So but in case of the revised IC for the Himalaya, it shows the MAT variation as per MAT variation, and it's a realistic one, and this data are the blue lines are 95% confidence interval of the reconstructed data. So we found that the the temperature is the most influential for Cichisica's distribution in the and their phytospectral differentiation in the Himalaya. Now to this we will end here, and we thank everybody for your patience, and I just want to show you my phytolith group. He is Professor Shubhi Bera, to whom I have learned everything. He is my mentor, guide, and teacher. And these are all the Dr. Deepak Farwa, Dr. Shantini Das, Ondila Vishwas, Dr. Madhav Najpal, and Vishwet Mukherjee are the phytolith team members which we work together to get it success. Thank you for your patient hearing. Thank you.
Thank you, Rubidi, for such a wonderful talk. Your lecture has enriched our knowledge about phytoliths and its implication on paleo environment. And I think it will be very much useful to all the students and researchers who have keen interest in paleobotany. And there are a few questions in the chat box which I would like to forward to you. Debjani Mahato has asked, Ma'am, how can we identify by looking from outside that we are taking samples of phytoliths from a place, since phytoliths are microscopic structures?
So you have to extract, not you cannot understand whether the sample you are collecting from somewhere will be producing phytoliths. So in that case, you have to select a proper place like forest floor or grassland condition, or if you are collecting samples from the surface, or if you are collecting lake deposit, then you have to find out the dried lake bed, or somewhere you have to be cautious while collecting your sample. Then you have to extract phytoliths in laboratory condition. There are different protocols I didn't mention in detail. So you can write me, I can tell you more about this.
Okay, and then the next question is also similar actually. It's from Thomas Otto. He has told that please mention methodology precautions that should be maintained while collecting pollen samples for dating.
For pollen sample, if you are collecting pollen samples for radiocarbon dating, then you should collect wearing a gloves, and you should take a sterile spatula or something like this, and you should have with you the Ziploc or aluminum foil with you, so that this after collection the samples immediately should be covered with this, and you should not touch the samples again, and just avoid the rootlets. Avoid rootlets.
Okay, next is from Orpon Pramanik. So question is, ma'am, is it possible to create an identification key for monocots or dicots by this phytolith study?
Yes, yes. I showed you a picture of Dr. Madhav Nashkar. He was working on the grass phytoliths of the deltaic plains of Sundarbans. He prepared and published a paper, I think, in Nordic Journal of Botany. You should consult that. And there is also another paper by Krishnan, I think 2009, published in the Linnean Society, Botanical Journal of Linnean Society. They have also prepared a key after studying 100 grass. But my objective is to know the environment better, so I don't want to focus more on the taxonomy. So these two papers can help you certainly.
Okay, there is another question from Munish Baloria. Can monocots and dicots differentiated by phytolith study?
Yes, yes, certainly. Because I showed you some slides where monocots are completely different in structure. They are having more diagnostic features than dicots. It can be differentiated.
The next question is there any group specific characters of phytoliths in grass species observed?
Yes, I told you in the lecture that for Panicoideae, they will be producing in abundance the lobate tiles, that means the bilobate and process. And for Floridoides subfamily, they will be producing the short saddle types. And for the Pooideae, they will be producing rondelles, trapeziforms likewise. There are some there are some markers for different subfamilies in the grasses. At least three subfamilies could be identified or distinguished.
Okay, now I think there is no questions actually, and I will request, we will send the email of the speaker. So if the participants have any questions, they can directly ask the speaker for further clarification. Okay.
Yes, yes.
Now, thank you, Rubiri, for being with us. Okay. Now I will request Ms. Rituparna Roy Chaudhuri to deliver today's vote of thanks.
Hello everyone. It's a great pleasure to me to propose vote of thanks to our resource person Dr. Abhishek Bhomik, guest faculty of Department of Anthropology, Central University of Odisha, Koraput. I would like to express gratitude to Dr. Ruby Ghosh, Scientist D, Birbal Sahani Institute of Paleosciences, Lucknow, Uttar Pradesh. Thank you to both the resource person for delivering such a wonderful presentation and make the webinar so interesting. With this, we have come to the end of day 1 of this 2 days virtual webinar on significance of paleo environment and its impact in its present scenario. I would like to express our warmest thanks to our principal sir Dr. Shunto Kumar Bose, Sri Rajarshi Mukherjee, IQAC coordinator and entire IQAC members, Shampoo Siddheshwari Mahavidyalaya for inspiring and guiding us. Finally, I would like to thank Dr. Shatarupa De, Head of the Departments of Botany, Shampoo Siddheshwari Mahavidyalaya, Ms. Shoumita Chatterjee, Head of the Departments of Anthropology, Sri Prasenjit Da, Departments of Zoology, Sri Animesh Manna, Department of History, and Ms. Shati Shet, Departments of Botany, and the entire technical support teams of Shampoo Siddheshwari Mahavidyalaya to organize the webinar so successful. Finally, I thanks to all the participants for joining us in this webinar. See you all tomorrow at 11 a.m. So stay tuned and stay safe. Thank you.